We now begin a series of lectures on metabolism and energy balance. And in this discussion or consideration, there are many different concepts that we're going to consider, and they will begin integrating several prior concepts and topics. So it really becomes more practical and interesting, in my opinion, as we go through this, because we all experience appetite and satiety issues. Do we eat too much? Is there enough to be eaten? And as Thanksgiving is coming upon us, we think about it in the sense of overdoing it and getting out of our energy balance and overwhelming our metabolism, and we may put on a little more body fat than what we started off with. So in that sense, I really enjoy this part of the series of studies of the digestive system and homeostasis. So we'll consider concepts related to the metabolic homeostasis and the control. And then ultimately, in this series, there will be at least two, but possibly three. I may just record the regulation of body temperature as a separate one, and the second in this series will focus more on the homeostatic control of metabolism with some carryover of metabolism in general. So we'll start with these first three in this presentation. So we're all familiar with appetite. Are you hungry? Do you find yourself in a situation where you'd like to be eating something? And everybody has different set points for appetite. As well as we are eating and our stomach begins to fill, which is of course going to happen, the actual gastric stomach, the anatomical structure that we're studying, then certain signals will be sent to the nervous system, and they will be both neural, primarily from the vagus nerve, and of course hormonal or endocrine, to tell us when we've eaten enough and we should stop, or we've reached satiety. So these are very important issues in the entire world as people are becoming more and more burdened by adipose tissue. They become fatter and fatter all across the world. More and more people are beginning to experience this as high-calorie foodstuffs become more readily available, and we eat more than what we actually burn in a day, so it becomes very important. However, food intake is carefully controlled throughout this whole system, and it seems too easily to be thrown out of homeostasis or reset at different points. So there are two competing behavioral states, appetite or hunger, that's the desire for food as we said, and the satiety or the sense of fullness or satisfaction to terminate the food-seeking and food ingestion behavior. Remember, appetite is or hunger is the desire for the food. In between appetite and satiety is food-seeking behavior, and then the capturing of that food, whether it's retrieving it from a refrigerator or the shelf or the local supermarket, and then preparing it and then consuming it. So there's all these different aspects dependent upon the somatomotor nervous system and the musculoskeletal system, quite obviously. So it's a very complex link between these two. However, from animal studies and from imaging studies in humans now with the non-invasive imaging techniques that are available, it's been found that the hypothalamus contains two key control centers, and these have been known for a long period of time. There's a feeding center and a satiety center. Now we know that we have known that they exist in this area for decades. But we still do not completely understand how they operate. We have a tremendous amount of knowledge on what happens when we're feeding or satiated, but we don't have a full grasp of how they are functioning normally and abnormally when homeostasis is at an imbalance. Definitely we don't know enough about how they are regulated to intervene when there is a metabolic imbalance. And I know you're aware that there are two sides of the eating disorders. There's not eating enough, hypophagia. Phagia, remember phagocyte eats stuff, right? So phagia means to eat. So there can be hypo or aphasia, my brain is not thinking of the word for the situation where people end up being skin and bones, and they look in the mirror and they still think that they're fat, and I'm sure you're saying, oh, that's boom, and thank you for remembering for me. And then, of course, there's the totally opposite side, the hyperphagia, or overeating, and the problems that are associated with that. So there are four types of input to the hypothalamus that we need to think about. There's the neural input from the cerebral cortex. There's the neural input from the limbic system. The limbic system actually is a link between somatic and visceral sensory and motor systems. So remember that is very important, and it links emotion to these deepest visceral functions, and obviously visceral with the GI tract is an obvious component to be expected within the limbic system. And there's a tremendous amount of peptide hormone signaling from the GI tract, but many of these neural circuits also utilize peptide neurotransmitters, and sometimes the same ones that are used for the signaling in the gut system. And then, of course, there's adipocytokines, signals that originate from the adipocytes, the fat cells themselves, and target other tissues, in particular the neural system, to coordinate body fat content with these neural functions that we've been talking about, neurobehavioral functions linking appetite with satiety. So there are two theories for the regulation of food intake, glucostatic theory and the lipostatic theory. The glucostatic theory proposes that blood glucose levels ultimately control the feeding and satiety centers. So as long as plenty of glucose is available for the neurons in the hypothalamus to keep operating, then the balance between feeding and satiety is equilibrated. There's no need or sense generated or motivation created to seek food, and the feeling of satiety is supported. The glucose drops, and you put the brake on the satiety feeling, and you put the gas pedal on for the appetite and feeding. The lipostatic theory proposes that the level of body fat regulates feeding and satiety centers. So we're all kind of aware of saying that, my body just seems to like this certain amount of fat on it, and I can't seem to get it to change its mind to go to a lower setting and stay there. I can diet and drop 10 pounds, but over a period of a few months, I kind of rebound right back to that. And then, of course, there's some truth to this, and it creates real challenges in trying to understand this system. It's a very complex system. Recent discovery of several peptides, especially leptin and neuropeptide Y, seems to support this theory of some critical mass of fat tissue within individual organisms, helping to set the whole balance for this homeostasis of feeding and all the behaviors in between and the satiety. You'd almost think that it would end up being something very simple, and on some of the levels you get very potent effects, but then on the other level, for humans in particular, you get is shifts toward imbalance. We all get fat so easily that it's an extreme challenge to try to capture and manipulate successfully. So the peptides regulate the feeding centers. So these things we certainly know, that there's neuropeptide Y generated at regions within the hypothalamus that connect with hypothalamic feeding centers, and that is a gas pedal. So it is a drive for the feeding. So if you inject neuropeptide Y into this hypothalamic feeding center, remember we talked about angiotensin. If you injected that into the supraventricular system, the animals would go right to their water bottle and just drink and drink and drink and drink until all that angiotensin 2 had been destroyed and the stimulus was removed. It's the same type of an effect you can get with the neuropeptide Y. Until the neuropeptide Y, every molecule has a half-life, so when you inject a certain amount, it's going to exist in the interstitial fluid for a period of time, and in the time period over which you can calculate that it would take that amount to be degraded, you get a very potent feeding behavior. So the animal will seek out the food in the typical location first, and then if there isn't food there, they'll go into a food-seeking behavior to find it if it's at another location, and then just commence with eating and keep going until the drug wears off. So there's an increase in food intake, an increase in the fat stores over a longer period of time. The temporal dynamics, as you know, are very important. You can eat one gigantic meal a week and then not eat anything else for the rest of the week, and for the few moments after you ate that big meal, you'll generate more fat, but at the end of the week, you're not going to be fatter. But if you eat that big meal every day, at the end of the week, you might weigh 3 or 4 more pounds, right? So this is the important way to think about these temporal dynamics all the time. Keep that in your mind of what might be an immediate, short-term, and a long-term condition that we're considering here. And then there will be an increase in leptin secretion. So this is an immediate response after a meal, but also over a period of time. So what's the average daily or weekly leptin plasma levels, and how does that affect the ongoing average activity of these neuropeptide Y neurons, right? And so there is a described feedback system. We don't know everything that this dotted line represents, but we know that there is a negative feedback system that exists. And then there are other neuropeptides and hormones, some of which originate in the periphery and are being exploited, like the GLP-1 that we talked about in the previous lecture, where certain analogs of that molecule are being developed to test in humans, either for the negative feedback for that one, and of course in cases of anorexia, finally remembered it, severe anorexia, we would be interested in drugs that would stimulate eating when the individual organism is completely resistive to food intake. And there's other conditions in humans where aphasia is induced. Aphasia meaning not eating. Anorexia nervosa is one example where aphasia is a symptom, but there are other symptoms and different causes versus, say, someone with terminal cancer and is not eating, but does want to be sustained. So we have need on both sides to develop and exploit these mechanisms. And then, of course, there's some that can impact the feeding center directly as well. and there's complex neural circuitry within the hypothalamus, some of which is understood, and then more of which is not understood yet. But we know that there's both a drive, a gas pedal, and a brake at all these levels, as well as the peripheral interactions of neuropeptides and hormones with the adipose tissue as the source of the leptin. So many peptides are known, as I said, to alter food intake. So we can just go through this list quickly of the peptide and the source. So they either increase food intake or decrease food intake. So ghrelin from the stomach, neuropeptide Y, as we said, from the hypothalamus, orexins in general. They're also called hypocretins from the hypothalamus, and they are secretions that impact food intake. And then those that decrease food intake, CCK, from the small intestine, and certain neurons within the circuitry of the limbic system and the hypothalamus that we just described. Leptin from the adipose tissue, olbistatin from the stomach, CRH from the hypothalamus. You recall that from the neuroendocrinology that we studied. Alpha melanocyte stimulating hormone from the hypothalamus. CART, cocaine and amphetamine regulated transcripts, so I guess you know how this gene was discovered, and inadvertently thereby found to be involved in inhibiting or decreasing food intake, and produced by neurons in the hypothalamus for this function. I state it that way because it may be expressed in other areas like reward circuits where it's more associated with getting addicted to these substances of abuse. But genes don't exist for us to get addicted to substances of abuse. They have other functions, and this one turned out to be associated with the ability of these two substances of abuse or drugs that we know will generate hypophagia. And then the glucagon-like peptide, the GLP-1 that I was telling you about from the intestines, so the analogs that are being developed that will activate the GLP-1 receptor are being developed and tested to decrease food intake to treat diabetes and obesity associated with hyperphagia. And then PYY is peptide YY, the amino acid sequence from the 3rd to the 36, so it's a pre-pro hormone that is cleaved into different fragments, and the cleavage fragment 3 to 36 amino acid residues of the parent molecule becomes an active hormone called peptide YY or PYY, and the source is the intestines. And this is a short list, so it's a very complex system. There's no simple intervention. Of course, if amphetamines were not addictive and associated with all the other problems that it generates, that's somewhat of an ideal drug, and of course had been used in the past for weight management, but is just overall there's too many risks, because it was very useful because it does decrease food intake, increases metabolic rate, increases physical activity, but not really in a healthful way if you are familiar with the effects of that drug. A lot of the motor behaviors induced by it are unproductive, and it has a negative effect on even human social productivity, like if at work and things like that, with long-term use. So it is, and then of course you've got the addiction and physical withdrawal and potential risks of cardiovascular catastrophes. So it's not ideal, and it is still kind of a baseline reference point for what we would think could be components of an ideal weight control approach. So it does fit into this energy balance idea of the key to the weight control. So if you can regulate energy input and the energy output, so in a case where you've got too much energy stored away, you would want to lower the left-hand side and increase the right-hand side, so that now you would be burning more energy than you are taking in, and your reserves would be depleted. Hopefully, you'll burn off the fat. But the long-term key is that you balance the caloric input with the caloric consumption, or the energy output. And of course, the caloric consumption internally, the energy output, it can either be work or heat. So there's really nothing else except for being stored as other molecules, and fat is the big one that we all think about. But of course, there's others that we'll talk about as well. So the three categories of work done by our cells, well, let's just back up. If we use the amphetamine as the example, it generates an increase in physical behavior. As we said, it's not always productive, but it increases skeletal muscle activities. So it impacts this, and then through that, but also through some other mechanisms, just generates heat. So this is how it contributes to increasing the energy output. So the three categories of work done by our cells is membrane transport, mechanical work, chemical work, which is the building of molecules, including the synthesis of energy storage molecules, such as the short-term energy storage, ATP. Long-term energy storage is glycogen and fat. So the glycogen is the other storage molecule that we needed to mention. When we continue on thinking about energy balance, there are methods for measuring energy use by an organism. One approach is direct calorimetry, which measures the energy content of food. And then there's indirect calorimetry that estimates the metabolic rate as a measure of energy use. So it can either look at oxygen consumption or carbon dioxide production, or the ratio of those two, called a ratio quotient, or the relative energy ratio. Did I get that right? Sorry. The textbook covers these in a bit more detail, and we'll leave it for that. What's important to note in the textbook is how they deal with the descriptions of the approaches to measure those quotients or ratios, and the association with a true basal metabolic rate versus a resting metabolic rate. What's the difference between the two? So the basal metabolic rate is the most common measure of metabolic rate, and there's 6 factors that affect the metabolic rate in general. Age and gender, the amount of lean muscle mass. In part, this is responsible for some of the gender differences. The level of physical activity, the energy intake, the diet, hormones, another that contributes to gender differences, and then genetic differences, how we metabolize certain genes, so certain molecules. So we all have a certain DNA sequence or genomics, and in some cases for enzymes involved in metabolism, we have different sequences, and based upon those sequences, there's different levels of metabolism that will occur. So kind of in a nutshell, that's the essence of the genetics. So there is some truth to the claims that an individual can have a low metabolic rate or a high metabolic rate, and that it is genetics. It's a little more sophisticated than what I had stated, but it is, in essence, related to the DNA sequence. Either the DNA that is involved in regulating the expression of the gene to produce that enzyme, or within the gene sequence, one of the exons, that would affect the activity of the enzyme, even though it might be present at normal levels, it might not be as active. So it's kind of a complex arena, but the area of nutrigenomics is built upon this final one. So if you're interested in that, just do a search on nutrigenomics. And you'll find a lot of very interesting information. So only the energy intake and the level of physical activity can be voluntarily changed. So otherwise, some type of nutritive supplement, typically, or drug will be utilized as an intervention to impact somewhere between the gene and the protein activity. And of course you can impact absorption and stuff like that. But anyway, let's move on. So two chemical forms of energy storage are the glycogen and the fats. The glycogen, as you should recall, is a highly branched polymer of glucose. So the stored glycogen binds water, so it's already hydrated. The textbook talks about consuming a handful of just raw sugar versus how much water you'd have to consume to hydrate all that versus how you can complex all that glucose into glycogen and have it pre-hydrated in a chunk of red meat and get the same amount of energy from it. So without having to consume liters of water. So the liver glycogen is used to regulate blood glucose within the same organism. Then the muscle glycogen is used to power the muscle contraction. So it is one of the reserves, local reserves, to meet demand once it exceeds what can be taken into the muscle directly from the blood. The triglycerides are the fats with the higher energy content per gram. So you should remember the triglycerides from the discussion of the digestion, the glycerol backbone with 3 fatty acids, the hydrophobic long chain fatty acids. And then the little water is required for fat storage because it repels the fat, and the energy in the fats is harder and slower to access. Excuse me. One point to bring to your attention here is that we still have in the formation of these two the same reactions to make and break. The reaction to make these polymers, and this is in essence a polymer, is to remove water. So there's the dehydration anabolic building up of these molecules, and then there's the hydrolytic cleavage of the bonds to break down the polymers into the monomers or the catabolic processes, and we'll talk about that a little more specifically as we proceed. But remember, water is generated to make these, and water is needed to split these into their smaller units. So metabolism is all of the chemical reactions in the body collectively. We extract energy from nutrients, we use energy for work and synthesis, and we store the excess energy. So there really are 2 types of metabolic pathways. The building up, which is anabolic. So everybody's heard of anabolic steroids. They build up the muscles. It's not the only thing that is anabolic, but it's typically the one everybody thinks of because it's the harmful effects of anabolic steroids and the fact that they're illegal in professional and even amateur athletic training has brought it to the forefront, so almost everybody knows about it. But what we need to know at this point is that it's only one of the anabolic pathways and only one of the anabolic compounds that's considered there. There are many other anabolic pathways. And then the catabolic pathways are the opposite, the breakdown. So the ones that I usually use as the examples are the anabolic steroids. They're androgenic, usually the male, testosterone type of a molecule that will target the buildup of protein and other molecules in skeletal muscle, as an example. So that would drive anabolic pathways to build up protein and other complex molecules in the skeletal muscle. And then using the same tissue, the skeletal muscle, a catabolic steroid would be like prednisone, steroidal anti-inflammatory medicines, or an anti-inflammatory steroid. There are, as you know, non-steroidal anti-inflammatory drugs, or NSAIDs, non-steroidal anti-inflammatory drugs, so NSAID, NSAIDs, just like acetylsalicylic acid and aspirin, or ibuprofen, and there's a few others. Some of them are prescription as well. So the NSAIDs don't trigger the catabolic events. So those are more favorable for long-term treatment. The anti-inflammatory steroids, which pretty much are the adrenal steroids, ultimately will cause a breakdown of the protein, especially in skeletal muscle, so they will cause skeletal muscle wasting. So the anti-inflammatory steroids activate the catabolic pathways in skeletal muscles. So that's a good example to think about two different steroids, one of which is anabolic and one of which is catabolic. But still, when you need a steroid treatment for the anti-inflammatory benefits, you accept the limited catabolic events that go along with it. So this is why you find that physicians are hesitant to prescribe the anti-inflammatory steroids. And if they do, it's for a short period of time and at the lowest dose possible. So the metabolism can be divided into two states. There's an absorptive, or the fed state, which is anabolic. So you just consumed all these things, you digest them, now you got to build them up to put them away, store them, or make the tissues that you need. So the post-absorptive would be the fasted state, which is going to be catabolic, where you turn to some of your stored materials and break it down, and use it for the energy that you need at the moment. So let's consider then the metabolic fates and the nutrient pools. So the ingested biomolecules have three fates. There's the immediate use and energy production, the synthesis into needed macromolecules, and the storage for later use in energy production. The nutrient pools are available for the immediate use, would be the free fatty acids, plasma glucose pool, of course, and then the amino acid pool. So hopefully you know that amino acids can be used to make glucose, and we'll talk about it anyway. And fatty acids can also be used to make glucose, but fatty acids can be used within cells, most tissue cells, to bypass glucose and just generate the acetyl-CoA that's used in the mitochondria, ultimately to produce ATP. And we'll summarize that in a few slides from now, after we have considered this. I really like the way that the authors designed this illustration, and we're going to look at each one separately as we go through a series of slides. But what I first want to draw your attention to is the glucose pool shown here. It is the most important, and it is regulated such that as that pool drops to lower and lower levels, there are mechanisms in place to protect different organ systems as we're going through this. And basically, first what you see is none should spill over into the urine. So the plasma glucose pool should never exceed what the kidneys can reabsorb, right? The pool provides the glucose to be stored as glycogen through the process of glycogenesis as part of the homeostatic regulation of this glucose pool. So we know that from the diet, our carbs are going to contribute to the glucose in the blood and come into this total pool. And if we consume too much of that, we know that through lipogenesis it's going to go toward the fats. But otherwise, we want to store it so we can get access to it a little more quickly by glycogenolysis. And then ultimately, if we're really having a need, or we're in a metabolic state like low-carb diet or zero-carb diet, then we're going to have to make glucose from whatever sources there are. But this range of normal plasma glucose, before any type of dramatic metabolic change is going to occur, is going to have a lower limit that is above the plasma glucose level required to sustain brain metabolism. So that's a very important aspect of this bucket of glucose pool to appreciate, is that it's actually illustrated here at thresholds to where you're going to trigger certain events. So the majority of the plasma glucose is used for the immediate energy production or is stored as the glycogen. So most of what we consume in the carbohydrate category is going to go straight to the glucose in the plasma, and the tissues that use it to generate glycogen stores will be doing that at a higher level right after a meal, and we'll get the majority of the glucose pulled out of the blood, so it's at least at the higher level within the normal range, and get it stored away the most readily available storage form. And of course, the glucose that is taken in by all those cells will also sustain the immediate ATP generation, and then whatever is coming in and then isn't flowing through that pathway to make the ATP will get shunted over to the glycogen stores. At the same time, some of the glucose is going to be feeding toward the lipogenesis, the production of fat. So it really depends on the amount of glucose, as we all know, flowing into the plasma glucose pool as to how much will be drained off toward lipogenesis. There will always be a little bit, but whether it'll be noticeable, to bring it to the terms of looking at our skin to see how much fat is under it, or our bellies, how big they're pooching out, that's what we're thinking about. Well, obviously, I'm a male talking about my belly, but everybody knows where they look for their their fat tracking behaviors. So the majority of the plasma glucose pool above that lowest limit to sustain brain metabolism is going directly to the metabolism in most of the tissues. And some of those tissues, of course, will be pulling off on the higher end to store it away as glycogen. So you notice that the higher, one of the first lines of protection of hyperglycemia is to store it away as glycogen. And then within the normal plasma range, it's probably somewhere around 60 to 100 milligrams per deciliter of blood. I might be corrected through the lecture series here, but that's a pretty good range to think about, to where the tissues will still be drawing from the plasma glucose to meet their metabolic needs. If it falls below that lower end, then the tissue metabolism will turn to something else, and the brain will still be safe for its glucose needs. So the free fatty acids from the diet are used for immediate energy production, or they're stored as fat molecules in the adipose tissue. So the free fatty acids and the glycerol, of course, can feed in to the lipogenesis process to form the fat stores, the neutral lipids, the tri-fatty acids. And the glycerol will be provided for the metabolism in most tissues. And if it isn't needed there, as with the free fatty acid pool, we see the free fatty acids coming down. If they are not needed there, if there's enough glucose as we saw before, then as excess nutrient they will be available for the fat stores. And then, of course, as we reach a point where total nutrients, the total number of calories burned in a period of time is less than the caloric intake, then we draw on those fat stores after the glycogen stores have been depleted. So we're all pretty much aware of that. We've got to reduce our daily caloric intake pretty low, to like 1,000 or less, really to get our bodies to turn to the fat stores and start removing from that to feed in to the energy needs for the tissues. Then the amino acids are used for building all the proteins that we need. So just think of every protein, every enzyme that we use, digestive enzymes and enzymes within the tissues and cells and stuff like that. There's a lot of proteins that turn over every day. Skeletal muscle protein might not be turning over, the actin and myosin, but still there is a rate, even though it might be a low level, a rate of turnover. Or if you're working out every day, weight training, there's a lot of damage and you need to repair the proteins that have been damaged. So that's the first place that the amino acids are going to go, is into the proteins. But from that amino acid pool, there's the protein synthesis and the total body protein. So it's every protein you can think of. Keratin is a protein, and think of how many dead cells you shed on the surface of your skin every day. I don't know if you've ever seen those things where they show somebody walking and all the dead skin cells that are falling off all the time. But anyway, so we're constantly losing protein just from the surface of our body, our skin cells. So keratin is a protein. That's our hair. Your hair grows every day, and that's protein that's consumed. It still might be there, but us guys try to shave it off every day, and it keeps growing back. So there's a protein that is turned over every day, and we have to continue to turn to our amino acid pool from our total body amino acid pool to grow our beard every day. Girls shave different body hair regions, and the growth rate isn't quite the same, but it's still every day it is growing back on the legs and the armpits if you shave there. So we need to think of that as an example of body protein turnover on a daily basis. So if that amino acid pool is more than what's needed to meet that demand, then it will be used to generate glucose to contribute to the glucose pool. But more so, what we are thinking about is that if the demand for glucose exceeds what can be contributed by glycogenolysis, gluconeogenesis from amino acids is one of the first means of producing new glucose by cells. And there's just a few cell types in the body, liver cell and skeletal muscle cell, that utilize this mechanism. The liver is the most successful. So this brings us to that summary of metabolism then. All the enzymatic systems, and there's thousands of genes, not hundreds of thousands. Well, we don't even have hundreds of thousands of genes. The human genome is about 24,000 genes or something like that. But there are 1,000 or more genes dedicated to this homeostatic metabolism, and more when you think of producing the other body proteins, the mechanisms that are required for protein synthesis are not necessarily included in this idea of metabolism when we're thinking of just about how much of the fats, carbohydrates, and proteins come in, and what we use for other purposes rather than just making fat. So in this context, then, we have to think of the fate of glucose, the biochemical pathways for the energy production. How do we produce ATP, and where do the other systems, like the fatty acids and some of the amino acids, the protein breakdown, contribute to this process? So there are interconversions of the glucose and the glycogen with this intermediate glucose 6-phosphate. So it is energized by adding the phosphate high energy bond to it. So this accomplishes a couple of things. It activates the glucose, so it's in this less stable form, and it's very likely to be converted to something else. One thing it can be converted to in the liver only, that they're saying here, and that's a safe conclusion to draw, is that it can be converted to glucose. So there's an enzyme that will convert glucose 6-phosphate to glucose, and it is expressed at the highest levels in the liver. And then, of course, there's in every cell the enzyme that will add the phosphate to the number 6 carbon on the glucose to generate this glucose 6-phosphate. And then in most cells, there's the capability to generate glycogen and then to break it down, and the glucose 6-phosphate is the form of glucose used to make the glycogen, and then is generated when the glycogen is broken down. So it is a glucose polymer, but when it is broken down, it's broken down to glucose 6-phosphate that feeds right into glycolysis. And as you might recall, there's some ATP consumed, obviously here, to get this phosphate on there, but a few other locations where the ATP is consumed, and then some is generated, and there's a net, the difference between what's consumed and total produced. There's a net of 2 ATP generated by glycolysis. So glycolysis is the anaerobic means for generating ATP. We don't consume any oxygen to do this. And the end product of this pathway is pyruvate. So as you know, if nothing happens to the pyruvate, it is converted to lactate, and the effect of your muscles not being in shape when you work out a bunch will lead to more anaerobic production of ATP, and the lactic acid will build up. So glycerol from the fatty acids can be converted to an intermediate within this glycolysis pathway for contributing to the ATP production and the production of the pyruvate. And then some amino acids can be deaminated and can be converted to pyruvate. We are obviously simplifying this it's more sophisticated. So if you read more detailed information, don't let it throw you off. We're trying to introduce the generalities here. The anaerobic conditions, as we said, contribute to lactate formation, and then the aerobic conditions will favor the direction of having this at a higher stable level of pyruvate. So this reversible reaction will be going on all the time. Under aerobic conditions, you're going to be drawing pyruvate into the mitochondrion, and so lactate, although it'll be produced, will be more readily converted back to pyruvate. It will not accumulate and will be drawn off into the mitochondrion. And in the mitochondrion, it goes through the tricyclic acid, it will be converted to acetyl-CoA and requires the coenzyme A. And after the acetyl-CoA has been produced, it'll be drawn into the citric acid cycle, or the TCA cycle, the tricytric acid cycle. And when it is drawn in there, of course, it gives up the CoA, coenzyme A, to be recycled and reused there. And then, of course, carbon dioxide will be given off, and 2-ATP at this point. So this introduces another aspect, although in the mitochondria now, not in the cytoplasm anymore, where other sources of energy can feed in, and this is where the fatty acids, so for the fats, the triglycerides, the fatty acids feed in here within the mitochondria to generate acetyl-CoA. So I mentioned that before. This is through a mechanism of oxidation, through a beta oxidation mechanism. So you may hear the term beta oxidation of fatty acids, and that's what it's referring to here, is that it is oxidized to produce acetyl-CoA, and along the way of along this pathway to the acetyl-CoA from the fatty acids, ketone bodies will be generated in the liver. So individuals that are on ketogenic diets are burning fatty acids through this mechanism. If it's low carb, then there's not much coming through this way. There'll be glycerol coming through, but the fatty acids will be burned at a higher level. Beta oxidation will be accentuated, and through the beta oxidation process to produce acetyl-CoA, ketones are generated. So that's why it's a ketogenic diet. But this is reversible, so under conditions of low carb, it can favor the beta oxidation to the acetyl-CoA. And then, of course, some amino acids can also be deaminated and then be converted to intermediates within the citric acid cycle. So anywhere that you feed in carbon, right? So one way of picturing all of this is the carbon flux. If they want to know what's happening to glucose in an organism, they can use a form that generates a signal, and sometimes it's radioactive. The earliest work was using carbon-14, of course not in humans, but anyway, in some animal studies. And so they could label different carbons throughout the glucose molecule and find out where those ended up, or in fatty acids, or in the glycerol, or in amino acids. So this is the way that these pathways were worked out. But you can read about the methods that are used in humans to track the utilization of glucose in this type of a situation, and they can determine certain intermediates, and it is non-harmful means of tracking labeled glucose. So anytime you feed in carbons, then you're going to drive the system from that point to try to return to homeostasis, And so if you increase the activity in the citric acid cycle, more CO2 and ATP would be generated, right? And then through the citric acid cycle, as you hopefully recall, you're going to get high energy electrons and protons. And of course, these are transferred to the electron transport system where when oxygen is present, that's where you generate the highest amount of the ATP, the 26 to 28. So we've got, say, 28, 30, 32 generated so far here from, of course, that's based upon a single glucose molecule or a single turn through here. So this idea of the push-pull control in metabolism is extremely important. And we've seen several of these reversible reactions. The majority of them are carried out by different enzymes. And what's important about this, rather than just having a single enzyme control it, is that you can control the enzyme to control the direction of, and you can control a specific enzyme to control the direction of the reaction. If it was a single enzyme at a reversible reaction, then it depends upon the concentration of the product B or the substrate A. If we're considering this is an earlier starting point, and this is a destination, right? So we have our reactants A and B. A is the substrate and B is the product. Enzyme 1 in this case is going to convert it to B. Enzyme 2 is responsible for converting B back to A. And so this gives us a more regulatable system, a system that can be more highly controlled and regulated. So there's no net synthesis of substance A or B when the degree of activation or the amount of active enzyme A is equivalent to the amount of active enzyme B. And I say equivalent because it has to be the activity that's equivalent, right? So you might have 2 times the amount of protein here, but if it's only half as active as that, then you have equal activity. I hope that makes sense to you, right? Anyway, to move on, if you somehow enhance the degree of activity of enzyme 1, and so you have a gas pedal, and at the same time you put the brake on enzyme 2, then you're going to favor conversion of A to B, or in this case, glucose to glycogen. So by having this local regulatory system at the level of the enzyme activity, when you have shortly after having eaten a meal, over the hour or two after eating a meal, the regulatory system is going to be set up like this. So you're going to put the gas pedal on the enzyme that's going to make the glycogen, or the enzymatic pathways, and the brake on the reverse system. And then several hours later, if you are now into the afternoon after breakfast and you didn't eat lunch, you're going to reverse that. You're going to put the brake on the enzymatic systems to convert glucose to glycogen, and put the gas pedal on the enzymatic systems to convert glycogen to glucose. So you'd have a net glucose synthesis. So they make me a bit disturbed here, because they start us off by saying enzyme 1 and enzyme 2, but as you recall in the earlier slide, they showed it was glucose 6-phosphate that's converted to glycogen, and glycogen is converted to glucose 6-phosphate. And then there has to be another system in place to convert the glucose 6-phosphate back to the glucose. So then I started talking about enzymatic systems that are getting the gas pedal and the brake here. So hopefully you recognize that transition of terminology that I used. We're not just talking necessarily about single enzymes. It can even be one or more enzymes in a particular pathway that'll get the gas pedal and the brake to reach these ultimate endpoints of either a net conversion of glucose to glycogen or net glycogen to glucose. And that's not the only pathway that applies to, but it's the one that is most relevant to our discussion at the moment. So the metabolism then, when we consider the fates of nutrients in the fed state, is shown on this table. So we think of carbohydrates that are absorbed primarily as glucose. As you recall, galactose and fructose were also absorbed, but that's fine. Right now we're just going to think of the glucose. The galactose and fructose ultimately have to be converted to glucose to be circulated and used. The proteins will be absorbed primarily as amino acids, and the fats primarily as the triglycerides. So the carbohydrates will be used immediately for energy through the aerobic pathways. We just saw that. Used for the lipoprotein synthesis in the liver, and for glycogen in liver and muscle. So the liver is going to take whatever glucose. Remember the liver for the digested and absorbed carbohydrates and proteins. They are going to be passing through the hepatic portal venous system first, and so the liver has first shot at them. And then for the fats, some of the free fatty acids will be transported in the blood directly to the liver, and of course the liver can generate the glycerol that it needs to take those free fatty acids right straight to the tri-fatty acid. And then the reassembled tri-fatty acids will reach the liver secondarily after having passed into the tissues. So the tissues will get first shot at the tri-fatty acids for local degradation by lipases and then uptake. And we know that that's mostly fat cells that'll be doing that. So I always want to remind you of the absorptive pathways. There's the split when it comes to the fats that are absorbed. Those that are absorbed primarily as triglycerides and stay triglycerides are going to go through the lymphatic system first. Remember, they're absorbed into the lacteal, and then to the systemic venous circulation, and then through the lungs, through the left heart, and to the whole body, and then ultimately what gets through the hepatic artery will get to the liver. But any free fatty acids that get directly into the plasma will go through the portal venous system straight to the liver. So when we see liver here and liver here, the liver got first shot at the glucose, any of the carbohydrates, before it was distributed systemically to the muscle. And hopefully you have looked at the anatomy of the liver, either in other courses or in the figure in the textbook. We didn't have it in the presentation. But you see how the hepatic portal system and the hepatic arterial system come together at the hepatic sinusoids, and so all the hepatic liver cells, the hepatocytes, are freely bathed in all the blood that comes to the liver, and so they have plenty of opportunities to remove from the blood what they need. So they really do get first shot, and then what is left, of course, should still be in the normal plasma glucose range for the skeletal muscle. So the reason that I take the time to talk about this so much is that we need to think about, when we end up talking about insulin, I like to present insulin as a molecule that turns our cells into sugar sponges to soak up all the glucose that they can out of the blood. And so the glucose transport in the liver is affected by insulin, but otherwise there's a baseline level of absorption because of this right here. It gets first shot, so it's got to have a constitutively or constantly active glucose transport to be the sponge to take it out of the hepatic portal venous system. And then skeletal muscle glucose uptake is going to be very highly regulated by insulin. But when insulin is present, both of these two will dramatically increase their sugar sponge capabilities, right? And if they don't respond properly to insulin, the sugar stays, the glucose stays in the plasma, and of course that's insulin insensitivity that is seen in the metabolic syndrome, the pre-diabetes, as well as into diabetes mellitus. And we'll have a separate talk about that as we proceed. So any excess then is available for the fat cells to take up and to store, but the liver also is going to store this. I'm sorry, I may have been talking about this as if it was lipid or fat synthesis, but lipoprotein synthesis is dependent upon the carbohydrates as well. And of course we're going to talk about LDL and HDL, the forms of cholesterol, as we proceed. The LDL is low density lipoproteins, so the lipoproteins are synthesized by the liver. Let's put that into perspective before we get back to the fat here. The liver also will store some fat, but it prefers to put the carbohydrates as glycogen, and it's only under extreme conditions that it'll start putting down a lot of fat, and of course that's not good. Fatty liver is damaging to the liver. And of course we typically will see it stored in the adipose tissue. So the liver may generate free fatty acids, but ship it out for the adipose tissue to take up and store. So the glucose is converted to pyruvate, to acetyl-CoA, and then to the fatty acids. So remember we talked about fatty acid beta oxidation to generate acetyl-CoA. Well, acetyl-CoA generated from glucose can be used through another series of enzymes to generate the fatty acids. So remember, that's a balanced system of making them and breaking them down, and you either drive it in one direction, so you're producing more fatty acids than you are breaking down to produce acetyl-CoA, or you're hardly producing any fatty acids and you're breaking down what is present. So there are those extremes. In other words, don't get locked into these linear pathways. Keep in mind that both directions are ongoing all the time. And if you have two separate enzymatic pathways, and there's a lot of enzymes involved in this and their genes to get from acetyl-CoA to the fatty acid, as well for the beta oxidation, there's a whole set of genes and the enzymes, proteins that are from those genes to reverse. So many different regulatory points for controlling this fat production versus fat breakdown. So the proteins come in as amino acids, as we know, and pretty much we need them to make proteins. And there's a lot of proteins that are made every day on an ongoing basis. Protein synthesis is always happening, as we said, so it's the first thing that we're going to expect them to be committed to. But if they are needed for energy, they will be drawn off. So in other words, the enzymatic systems within a cell that can convert the amino acids to the intermediates will be activated or increased. And if they're more active, then whatever amino acids are there are going to be competitively drawn out of that pool to be converted. So protein synthesis can be reduced simply by increasing everything that you need to draw the amino acid pool from inside a single cell. Think of it that way. In this case, a hepatocyte. Out of that's available for protein synthesis in general, and you're drawing it into the intermediate pool for the aerobic metabolism that we saw before. And then excess amino acids will be converted to fat and stored in adipose tissue. So the amino acids ultimately can be broken down and fed into the pathway such that acetyl-CoA will be produced at a higher rate, and then if that's the case, then that acetyl-CoA is going to drive and favor the fatty acid synthesis pathway. And of course the fats will very happily just stay as fat and get stored. So the liver, as we said, will produce or store fats, but it tries to keep that at a minimum, so our fat cells are usually more than happy to take it up and assemble it into the stored form. So let's take a look at the transport and the fate of the dietary fats that we had been talking about then. So we notice here that there's a lot of systems represented in this cartoon with a focus on the intestinal cells, but a lot of systems just generalized in this black box kind of an approach. But we do see the green lymph in between the chylomicron and the blood, and then the free fatty acids directly into the blood. So that's what we were saying before. So these will go to the liver first, to the hepatic portal system, and then those in the chylomicrons will be taken into the lymph and then get into the blood before they get to the liver and be acted on by the liver. So in other words, the blood being shown here for the free fatty acids will go to the liver first, then back ultimately to the blood, and then be available for this. But ultimately, an hour or two after you've eaten and you've absorbed all this, there's this mixing of everything. So what's in the plasma at that time has already circulated throughout the body several times. So if you're talking about looking for fatty acids in the blood a very short time period after they've been absorbed, most of them are straight from the absorption here. After having gone through the liver. And then later, it's all been mixed through the whole system. Okay, so the monoglycerides, the phospholipids, and the free fatty acids, as you recall, were absorbed, and then the apolipoproteins are important for the assembly of the chylomicrons, and the cholesterol comes along with it. And then the chylomicrons in the plasma are acted upon by the lipoprotein lipases. And the key is down here in the bottom right. I don't think it goes through that in a series. And then the CM remnants, as you recall, are taken up by the liver. The HDL cholesterol is going to be retrieving cholesterol from the plasma or cells throughout the body and returning that, if you want to think of it returning, or delivering it to the liver to be metabolized to produce the free cholesterol, the free fatty acids, and the lipoproteins. And the cholesterol is acted on to generate bile salts that are secreted through the bile duct. And those bile salts, as you recall, are involved in contributing to the digestive process, and the cholesterol is returned to the system. So any dietary cholesterol contributes to an additional cholesterol coming into this system. Substances that prevent cholesterol absorption either block its transport or absorb it so that there's less available to be bumping into the surface of the intestinal cells, will remove cholesterol from this total pool and lower, ultimately, your plasma cholesterol levels. The lipoproteins from the liver are going to be secreted by the liver in the form of a low-density lipoprotein, and they will have cholesterol in them. And this will deliver cholesterol to most cells cholesterol for synthesis. I don't like the way that they're wording this here. So most cells will reduce their own cholesterol synthesis if the cholesterol is delivered to them by the liver by the low-density lipoprotein cholesterol. But if there's too much of this because of our dietary cholesterol, or if the liver synthesizes too much cholesterol to be put in to here, then you'll have very high LDL cholesterol levels, and it's more than what cells need, so they're shutting down their own synthesis and taking up what they need, and then there's still far excess LDL-C in the plasma, and of course that's what ends up in the macrophages in the wall of the blood vessels and leads to the atherosclerosis. You'll have to refer to the vascular lecture for that review. Hopefully you're remembering the foamy cells and the fatty streak and all that stuff. That's from the excess LDL. It's more than what the cells need to maintain the cholesterol in their system. So it stays and becomes oxidized and then gets through the wall of the blood vessel and causes all those problems. So the textbook goes through a very nice discussion of the HDL-C versus the LDL-C. We'll back away from that for a moment to follow the glycerol from the chylomicrons after the LPL acts on the trifatty acids and breaks down the chylomicron, and the free fatty acids that have gained access to the plasma after going to the portal venous system in the liver and then into the systemic venous system, then are floating around in the arterial blood and are available to the adipose cells for uptake to reassemble as the triglycerides that are stored as fat droplets in our fat cells. And then at the same time, there's some level of activity of the lipases within the adipose cells, and this can be regulated up or down. And so there's some level of lipolysis putting these back into the plasma. So this is a cycle that is ongoing, and if it is at an imbalance where there's more of this going to the triglycerides than coming out, we accumulate fat within our fat cells, right? So one final comment before we leave this. HDL cholesterol represents not only the delivery of the reabsorbed cholesterol or dietary cholesterol coming to the liver, but it also represents the cholesterol that the HDL picks up from the cells. So each body cell uses cholesterol in its membranes. So hopefully you remember the phospholipids and cholesterol are found within membranes, right? The lipid bilayer. Well, every cell loses cholesterol from its cell membrane on a constant basis. It's there, and everything favors it staying in the cell membrane, but it will be lost. And then there's some proteins that will move cholesterol out of cells too. So there's a cholesterol efflux, and there's an influx and an efflux of cholesterol, kind of like getting new cholesterol and getting rid of the old. And the HDL is responsible for picking up that cholesterol that comes out of the body cells and brings it to the liver to be metabolized as bile salts. And of course, this is not always a 100% return of what is dumped. Some of the bile does end up in the feces. So a healthy balance between the LDL and the HDL is important for normal function. So in other words, it's not good physiologically to have zero LDL, it's good to have your LDLs very low. And of course, that is because of the correlative evidence in human epidemiological studies that LDL-C is taking the cholesterol from the liver to most tissues, meets a critical need by the body cells, but that need is satisfied at a certain point. And when we exceed that point, LDL cholesterol, as we see increasing on the x-axis of this graph from 40 milligrams per deciliter up to 190 milligrams per deciliter. As it exceeds what the cells will need, it's going to accumulate and become oxidized and be associated with problems. So it increases the risk of atherosclerosis. Here we see on the y-axis the relative risk for coronary heart disease. And many drugs try to lower cholesterol levels by changing its metabolism. So there's a whole discussion that can be presented on that, and refer you to the textbook or other interesting reading to come to understand that better. So I want to return to this graph for just a moment before we consider this final point. In the textbook, there's a question that asks at what concentration of LDL cholesterol do you see the change in the negative effect on health, or the highest change in the relative risk of coronary heart disease? And this brings us back to the idea. I'm going to go to a felt tip here, and I've tried this on other slides, I remember, where we do the tangent to the curve, right? I'm not getting these very good, but I think you can see if they were doing a straight line, when would the slope of that curve, and I exaggerated it here, the change in y over the change, no, this is the change in y over the change in x, when will the slope of this curve be greatest? And of course it's out here. If I was asked this question on an exam, I would say 190, because it's a number that's on the scale, but it's somewhere between 160 to 190 that you see a really dramatic increase in that risk. And so that's how the little bit of calculus, the line that's tangent to the curve will have a slope, and the more positive that slope, then the higher the change in the rate of the relative risk of disease will be associated with that curve, that portion of the curve. So this is why if someone has LDL above 130, they try to get it below that, so that you're in a better range for the risk, and then you continue to work on it to get it down into the lowest risk range. Now, low HDL is another risk factor for atherosclerosis. So I'm just going to go back to this one to make that point again. So too much LDL-C, as we saw, is going to leave too much around in the plasma to cause problems, to become oxidized. And once it becomes oxidized, it can't be taken up by the regular receptor mechanisms by cells and destroyed. So it's taken up by scavenger receptors of the macrophage, and then the macrophage can't degrade it, so it accumulates and they become foamy cells, and then you get the fatty streaks and things like that. So the accumulation is bad. So that's how the LDL-C becomes harmful as it gets to higher and higher levels with the homeostatic imbalance. If you don't have enough HDL-C to pick up excess cholesterol, and bring it to the liver, then you still have excess cholesterol around, and it can become oxidized and cause problems as well. And so the little bit of LDL-C that would be present is, relatively speaking, going to have more bad cholesterol with it to cause problems. So in other words, clinically, it's not favorable only to try to get LDL down. You have to take steps to get HDL increased if it's low. So it's the ratio of the two that is becoming more of an emphasis clinically. And again, I refer you to the text to see their very nice discussion of that. Well, now let's consider the fasted state metabolism to see what happens in between meals. So first, the liver glycogen is going to be broken down. And of course, this is not the entire glycogen reserves. It's some of the liver glycogen is going to be broken down. So remember, it's constantly being made and broken down. So the anabolic and catabolic processes are constantly ongoing. And you regulate them, as we saw on that one slide, to either favor glycogen production or glucose production from the glycogen. And so that's what we're going to see happening during the fasted state. So whatever you ate contributed to the blood glucose and free fatty acid plasma levels, and those were used for the energy needs at the moment, or the cells stored away. And as time goes by, the cells are going to decrease their own stores and place a greater demand on plasma glucose, and then the liver will be told, as you know, by glucagon, to put more glucose into the plasma. The adipose lipids will become the free fatty acids and the glycerol that enter the blood. So there will be a slight shift in the fasted state for the triglyceride stores to be broken down into the free fatty acids and the glycerol. So remember, this is a reversible process as well. So in between meals, there is a shift to where there's more triglyceride being broken down into free fatty acids and glycerol, but it is not to such a high degree that we're going to lose a significant amount of fat, maybe not even the amount of fat that we just put on from the meal. And this, as you know, is how incrementally over time fat accumulates. That in between meals we do turn to the fat. The fat cells are stimulated to break down fat, but it's never to the degree necessarily that they just built up fat reserves, because the liver has plenty of glycogen to contribute to the glucose, plasma glucose levels. And of course, it needs to make its own glucose 6-phosphate from glycogen through glycogenolysis to meet its own energy needs. Right? So it has to take care of itself first, as well as meet the needs for glucose in the plasma. So it will turn to its minimal free fatty acid reserves for beta oxidation to generate glucose through gluconeogenesis. But typically, that will be balanced very nicely so that you don't get a total depletion of the small amount of fatty acid reserves in the liver, because the free fatty acids will be coming from the fat cells. And so in the between meal fasted state, this is the smallest component. And under extreme conditions, to where it's an extended or long-term fasting, and then, you know, the definition of fasted, long-term fasted, and starvation varies. Some people say starvation starts at 24 hours or only after 2 days or 3 days of fasting. This transition from an acute fasted between meals to a long-term fasted to a starvation state is something that requires some thought as to exactly what criteria would you be using to differentiate between them. We often say, like, I'm starving, but that's not really what we're talking about. We're talking about the between meal fasted state. But if you, as you know, some cultures truly do fast throughout the entire day, or some religions have certain days where there's fasting that occurs. And so you will get a pretty robust shift to the free fatty acids and a certain degree of ketone body production. And so next we add the skeletal muscle, and so on a mass per total body mass basis, as you know, most people have more skeletal muscle than the mass of the liver, and it varies. Sometimes I wonder what my ratio of skeletal muscle to body fat is. Anyway, we all know we just grab our body parts we monitor and get a feel for what this ratio is. But anyway, the skeletal muscle represents a tremendous mass of tissue in any human organism that has protein content and glycogen content and energy needs. And so it contributes to energy consumption, but it can also contribute to the essential needs for glucose for the whole body. So in other words, you can get muscle wasting to contribute to gluconeogenesis to provide glucose for all body cells in cases of starvation, right? So there's this transition. When exactly do you call it starvation? When you got a significant burning of fat and a significant burning of proteins, degradation of proteins in the skeletal muscle, for its own energy production, as well as to contribute to plasma levels of amino acids, or the anaerobic process of breaking down any glycogen. In starvation, this glycogen should be depleted, right? And we would expect that most, if not all, of the liver glycogen is going to be depleted. And then you're relying on all aspects of gluconeogenesis to maintain any type of plasma glucose concentration in this transition from a short-term fast to a long-term fast to a starvation to extensive starvation, of course. So the muscle glycogen can be used for energy. Muscles also use fatty acids and break down their proteins to amino acids that enter the blood. So again, all these systems are operational at all points in time, and it's the shifting through regulation of proteins within the metabolic pathways and the genes responsible for those proteins being regulated under certain conditions of fed versus fasted, and what degree of being fed and what degree of being fasted. So try never to think of them as one particular state. It's always dynamic, but there is going to be a basal level of activity upon which is superimposed a dynamic process. The brain requires predominantly glucose, and as we saw, remember its level of plasma glucose was the lowest before there would be any kind of a disaster. And as it approaches that, as plasma glucose levels approach that low threshold for the brain, typically you're going to trigger the mechanisms for fat burning, and ketone bodies will be produced, and those ketone bodies can pass the blood-brain barrier, and then the enzyme systems are available in the brain to convert them into predominantly the acetyl-CoA to feed into the same energy production system that the glucose ultimately does after it's been broken down to the pyruvate and then pyruvate to the acetyl-CoA. So acetyl-CoA is a very important molecule shared by many, many different systems, and it's one of the metabolites for which there is the highest level of competition by metabolic pathways. But energy comes first. So most everything else will be challenged or shut down to favor the energy, the ATP production.